Hello. So in this third and last part of today's lesson, we are going to talk about the derivatives of the endoderm. So what is formed from the endoderm? The, the endoderm is responsible for the formation of many tubes or tubular structure in the adult body of the animal, of the vertebrate, however, not the muscles. What does it mean? It means that the endoderm form most of the internal organs. But what does it form? What is responsible for the formation of? So the endoderm is responsible for the formation of the digestive tube. So, and the stomach, for example, is formed from the endoderm, or the digestive tube, the intestines. The endoderm is also responsible for the formation of liver, gallbladder, and pancreas. The endoderm form also the respiratory tube, which actually derive directly from the digestive tube. So it is a budding out of the digestive tube. So the lungs is derived directly from the digestive tube. So lungs and pharynx are also derived from the endoderm. And finally, many glands of the body, including tonsil, thyroid, thymus, and parathyroids, are formed from the endoderm. So during initial development of the embryo, we know that the visceral endoderm is substituted by the real endoderm, the definitive endoderm. However, how do the cell that migrate from the external part of the body, so from the epiblast, they are migrating inside the primitive groove, and they are going down and becoming initially the endoderm and then in the middle the mesoderm. How the cells are differentiated in between the endodermal cells that are internal and the mesodermal cell in the middle. Well, this is mostly due to expression of SOX17 or brachyury. Brachyury is expressed in mesodermal cells. So the cells that are migrating from the epiblast inside the primitive groove and then become mesoderm are expressing brachyury. And brachyury is driven by expression of BMP and FGF in those cells. However, the cells that are migrating and becoming endoderm are going to express SOX17. And the expression of SOX17 is driven by nodal. So expression of nodal induced expression of SOX17 in the definitive endodermal cells, while brachyury is expressed in mesodermal cells driven by BMP and FGF signaling. Now, along the anterior-posterior axis of the embryo, of the developing embryo, the endodermal cells are going to develop in different tissues. Which kind of tissue they are going to develop into depends on the level of expression of BMP, Wnt, and FGF signaling. And we already know, we talked about this during the anterior-posterior axis formation lesson, we already know that along the anterior-posterior axis of the animal, we have a gradient of expression of BMP, Wnt, and FGF signals. In fact, we have higher expression in the posterior of the body, while we have lower expression in the anterior of the body. In fact, the endodermal cells will, in the anterior of the body near the head, will develop into the anterior foregut cells, which are the precursor of lungs and thyroid. In the posterior part of the body, the endodermal cells are exposed to high level of BMP, FGF, and Wnt signaling, and will develop into the midgut, hindgut precursor cells, which then will develop into the intestine. So low level of BMP, FGF, and Wnt signals in the anterior part of the body will instruct the cells to become lungs and thyroid, while in the posterior part of the body, high level of these signals will instruct the cells of the endoderm to become intestine. Middle level of expression of BMP, FGF, and Wnt signals will instruct the cells of the endoderm to become posterior foregut precursor cells, which are the precursor cells for pancreas, excuse me, pancreas and liver, hepatoblast. Hepatoblast are the cells that will become liver. So, of course, the endoderm not only form the intestine, the stomach, and the lungs, but also form the accessory organs. So just below the stomach, or if you want, caudally to the stomach, the endoderm buds out and form the liver, the pancreas, and the gallbladder buds. In particular, the hepatic diverticulum. The hepatic diverticulum is a big bud of the endoderm and form the liver, expand. So this endoderm will expand inside the inductive mesoderm. So there is an interaction again, as we saw for the kidney. We have an interaction between, in this case, the endoderm and the mesodermal mesenchyma. We have an interaction between the two, and the endoderm branch out and form different tubes inside this inductive mesenchyma, forming the mature liver in the adult. A portion of this hepatic diverticulum buds out, forming the gallbladder, while two pancreas buds that are called the ventral and dorsal pancreatic bud are forms on both sides initially of the digestive tube, of the endodermal tube if you want, and then migrate together to the left side of the body of the animal, and they fuse together, forming a single pancreatic glandula or organ. Now, it is interesting to note that in some vertebrates like us, the initial two tubes that connect the two part of the pancreas, the dorsal and the ventral part of the pancreas, to the main digestive tubes persist. So in the adult animal, we will find two tubes connecting the pancreas organ to the digestive tube. In other animals like us, only one of these two tubes persist, or if you want, the two tubes fuse together to become a single tube that connect the pancreas to the to the digestive system. However, in dogs, for example, two of these tubes persist. Some animal develop in doing one thing, some animal develop in doing another thing. Now, how is the liver specified? How is the liver specified? So the liver is specified from the endoderm, of course, but only in a specific region along the endoderm. And indeed, the only endoderm that is able to produce the liver diverticula is the one exposed to the cardiogenic mesoderm. So in this cartoon, I think we can appreciate how the cardiogenic mesoderm, or if you want the developing heart, is in an anterior position of the body of the animal. So in this cartoon represents a section, an anterior to posterior section of a vertebrate embryo. We can recognize here the head and the tail. In blue here is the ectoderm. We have this red structure is the notochord, and the yellow here is the endoderm. You remember here the endoderm goes around also the yolk sac. In some, or the initial endoderm goes around the yolk sac. Now, anteriorly, the mesoderm is forming, the lateral plate mesoderm is forming the heart. So anteriorly, the heart is inducing, signals from the heart are inducing the endoderm or the adjacent endoderm to produce the hepatic diverticula. However, the rest of the endoderm cannot produce the hepatic diverticula because of signals from the notochord. So the notochord inhibit the endoderm to produce the hepatic diverticula, and the hepatic diverticula can only be produced from signals from the developing heart. So what are the signals? Probably the signals are FGF signals from the developing heart. So the cardiogenic mesoderm is expressed in FGF. Do you remember that we were saying that BMP signaling from the endoderm are inducing FGF expression? Therefore, the cardiogenic mesoderm can develop. Indeed, the cardiogenic mesoderm express itself FGF signals, and these FGF signals are important because then they induce the endoderm to develop the liver. Not only FGF signals are important, but also BMP signals from the lateral plate mesoderm. So both FGF signals and BMP signals are important for developing the liver from the endoderm. The hepatic cells, so if you want, the hepatocyte are then promoted, or if you want, they grow because of other signals, including glucocorticoid hormones, hepatocyte growth factor, and Wnt signaling. So these are other signals that are important to promote the overgrowth, if you want, of the hepatocyte of the liver cells. Now, what is interesting here to notice is that the development of the pancreas require exactly the opposite signals of the developing of the liver. So the pancreas is inhibited by the notochord. The pancreas is induced by the notochord and it is inhibited by the cardiogenic mesoderm. So while the hepatic diverticula, if you want, the liver, is induced by the cardiogenic mesoderm, the pancreas is inhibited by the cardiogenic mesoderm. While the liver was inhibited by the notochord, the pancreas is induced by the notochord. So the pancreas is require the opposite signals compared to the to the liver. And one thing that is particularly interesting in the case of the pancreas is that the notochord repress SHH in the endoderm via FGF2 and activin. So, but the important thing to remember here is that the notochord, in this case, is repressing SHH signals in the endoderm. This is extremely strange because we should know, you should know, we know for sure that the notochord is in general a region of expression of SHH. If you recall, even when we were talking about the development of the neural tube, we're saying that the notochord is expressing SHH, and SHH is inducing the formation of the neural plate. So now here, in this particular case, notochord is repressing SHH, which is extremely strange. So SHH is in fact expressed in all endoderm, but in the region where the pancreas is formed. And this repression is particularly important because in this region where SHH is repressed, the endoderm become responsive to another couple of signals that come from the blood vessel endothelium. So the blood vessel endothelium express PDX1 and PTF1A. So PDX1 and PTF1A, and the endoderm is usually not responsive to these signals unless we remove SHH. So in this region, SHH is repressed, and therefore the endothelium, the endoderm become responsive to these signals from the blood vessel. And here you can see in this cartoon that the gut, which basically is the endoderm, become responsive to this signal from the aorta and the right vitelline vein, and then it buds out. And if you remember the picture that I just showed a couple of slides ago, the pancreas is formed on the two sides, if you want, of the gut, and this is because this region responds to the signal from aorta and the vitelline vein that are, of course, on the two sides of the gut. Finally, we're talking about the respiratory tubes on the lungs, the formation of the lungs. Believe it or not, the lungs cavity are formed initially from the same tissue that are forming the gut, and in particular the foregut. Even if the lungs, of course, are not related to digestion, the tissue itself comes from the foregut. So in the anterior part of the developing animal, the foregut at one point is divided in two. So there is what is called the laryngotracheal groove. This laryngotracheal groove separates the foregut in two regions. One that will develop in the pharynx and the esophagus, and one will develop into the trachea. This tracheal tube will then bifurcate and form the two, forming the two lungs buds. Now, it is important to notice here that again there is an interaction between the endoderm and the surrounding mesenchyma, enable this tissue to become either trachea and then lungs, or esophagus, and therefore keep going with the digestive system. So in a more dorsal position, so we are here anterior, okay, to the animal, but now we are dorsal, so toward the back. So in a more dorsal position, we are talking about this region, the mesenchyma produce Wnt inhibitors. So we are talking about this picture here. The mesenchyma produce Wnt inhibitors, this SFRPs, and these Wnt inhibitors block Wnt signaling in the endoderm. So when this Wnt signaling in the endoderm dorsally are inhibited, the epithelium become esophagus. So become the esophagus that is in continuation with the digestive system. However, ventrally, so we're talking about this region here now. Ventrally, we don't have the expression of these inhibitors in the mesenchyma, and therefore Wnt signals can induce the endodermal cells to form lungs and trachea. And in fact, when there is no these SFRPs signals, the epithelia become ciliated. So the trachea and lungs epithelia become ciliated, form cilia, which are typical of the respiratory epithelium. While the esophagus do not have the cilia, the lungs have obviously the cilia, and they will actually develop into bronchs and bronchioli later on, which allowed respiration in, of course, arthropods that live outside water. Okay, so the lesson is finished, and I hope you enjoy it. It is very interesting, in my opinion, how there is interaction between different tissue that allow the formation of the kidney in one case, or in this case, the formation in the last slide, the formation of different epithelia that distinguish esophagus and trachea. It's also very interesting to me to notice how liver and pancreas are almost mirror image of each other in terms of expression requirement to be developed. I hope that at the end of this lesson, and when you finish revising, you will be able to discuss and explain how the intermediate mesoderm develop kidney, and how heart and circulatory system are formed from the lateral plate mesoderm, and of course explain how the digestive tract and the lungs are derived from the endoderm.